bringing up a very common question that patients have is, number one, how did I get this? And number two, can I pass it down to my kids? And the answer is actually quite complicated. And so in these slides, want to try to sort of shed some light on it. First, just to sort of get some basic biology of how do we actually inherit diseases. So I tried to make a simplified version of this. Of each of our genes, we have 2 copies in each cell. So each of your cells has 2 copies of JAK2, for example. And when 2 people have a baby, they pass down, the mom passed down 1 copy of a gene and the dad passes down 1 copy of basically selects for one of the 2 copies to be passed down to each of the children. And so in this picture, I meant the red to be a bad mutant gene and the green to be good. So in this case, the mom had 1 bad copy and 1 good copy, and she gave the bad copy to her baby, and then the dad gave because he only has good copies, the good copy to the baby. And so this can be a little more complicated in whether or not to get a disease, you need 2 bad copies, which would be call a recessive gene, or dominant. If you only have 1 bad copy, then the baby gets the disease. Another complication is a thing called penetrance, which that not 100% of people who have a bad copy of a gene will manifest with the actual disease. So you can say that there's a lot of sort of gray zones about having a mutation and whether it causes a disease, but this is sort of the classic inherited pattern, which is not necessarily the case in what we call myeloproliferative neoplasms. So in a myeloproliferative neoplasm, you've always acquired the mutation in a blood stem cell. So it's theoretically the mutation itself is not passed down to your children. There's never been a human that has been described with a JAK2 V617F basically inborn mutation. Interesting, other similar JAK2 mutations, for example, a JAK2 V617I mutation has been described in some families, but they don't have MPN. They have a different type of a disease. But there is, so even though your JAK2, I'm just saying JAK2 because that's the most common MPN mutation, but the same applies for things like calreticulin or MPL mutations. Even though the JAK2 mutation isn't passed down, there is a very strong familial predisposition to acquire a myeloproliferative neoplasm. Many of you in this room probably have other relatives with a myeloproliferative neoplasm or other blood cancers. So first-degree relatives of MPN patients have about 4 to 5-fold higher risk of MPN. About 10 to 15% of people with MPN will have a first-degree relative with MPN, and that's a very rare disease. So you can see how strong this familial predisposition is. The JAK2 mutation is always somatic. So these are some more vocabulary words that you may encounter when sifting through your bone marrow biopsy results and things like that. So the word germline or inherited means the classic sense in that you got the mutation from the sperm or egg from one of your parents. Or somatic mutations means that it was acquired in a cell in your body after the sperm and egg came together. And it could be acquired any point basically during the lifespan. So in a myeloproliferative neoplasm, you acquired it in a blood stem cell. So you didn't acquire it in a skin cell or a gut cell. It was acquired in a blood stem cell that was in your bone marrow. Not in these slides, but another interesting question that people ask is, when did I acquire my JAK2 mutation? And we now know that people acquired their JAK2 mutation about 30 years prior to their diagnosis of an MPN. And in some cases, that can even be tracked back to in utero, so even before they were born. But they didn't actually get it from their mom. They acquired it in their cells as they were growing as a fetus. So there are truly inherited forms of diseases that look like MPN, but these are not MPN. These are called hereditary erythrocytosis and thrombocytosis. So there are families in whom through an inherited mutation that came from their parents, they have high platelets or high red cells. And this, I want to be clear, this is not considered a myeloproliferative neoplasm. The word myeloproliferative neoplasm means you have too many cells, and the word neoplasm means cancer. And to have a cancer, you need an acquired mutation. So these cases of hereditary erythrocytosis and thrombocytosis do not meet the criteria of a neoplasm. And interestingly, some of these mutations that are seen in these hereditary forms are very similar to the mutations that we see in a myeloproliferative neoplasm, such as mutations in the erythropoietin pathway. JAK2 is downstream of the erythropoietin pathway. Sometimes people can have mutations in MPL. I don't know if anybody in the room has a MPL mutated ET or myelofibrosis. That's the third most common mutation seen in myeloproliferative neoplasms. That can all, similar mutations in that same gene, but in different locations, can be seen in some familial thrombocytosis or familial high platelet count. So I want to just clarify that probably no one in the room has one of these congenital diseases, but if you do, it's important to note that it's not an MPN. It's a different type of disease. And then on the other hand, there can be many non-MPN causes of having high blood counts. Just because you have high blood counts doesn't necessarily mean that you have an MPN. There are actually many more common reasons why people can have high red blood cells, or what's the scientific term erythrocytosis, such as taking testosterone, sleep apnea, smoking. Some medications can cause elevation in your red blood cells. Rarely can have this EPO-producing tumors that make your body basically produce too many red blood cells. High platelets, or call the fancy word thrombocytosis, a chronic inflammation. That's our body's normal response to sort of a inflammatory insult will be to rise our platelets. Iron deficiency is also another very common reason to have high platelets, and rarely solid tumors can result in high platelets. High white blood cells are called leukocytosis. Again, if you have an infection, our body's normal response to an infection, our white blood cells fight infection. So if you have an infection, your white blood cells are going to go up to help you fight the infection. Other non-MPN blood cancers, such as CML, can cause a high white count. Also medications, for example, if you're on prednisone, your white count's going to go up. So there's many. Just want to emphasize that high counts does not necessarily equal a myeloproliferative neoplasm. And on the other hand, does a JAK2 mutation mean you have a myeloproliferative neoplasm? Well, not necessarily. So in this study done a few years ago by Hans Hasselbach's group, basically screened, they have banks of blood from Danish citizens, and basically screened all the citizens for JAK2 and calreticulin. And they found, this is using a very sensitive test to really just detect very minute numbers of blood cells with JAK2 mutation. This is the JAK2 V617F mutation. They found that about 3.2% of all Danish citizens, if you screen their blood, will have a JAK2 mutation. And the yellow part, that's just the little top of the part, those are the people who actually did have a known MPN. But all of the gray part, most of them did not have an MPN. So they were just walking around fine, and they were just harboring small numbers of JAK2 mutant cells. And then calreticulin, there are some normal controls with calreticulin, but that number is smaller. Interestingly, this phenomenon is now becoming well known and common in our aging population. That previously we thought that if somebody has a mutation in their blood that's associated with leukemias or other blood cancers, that must mean that they have a blood cancer. But now we know that actually upwards of 20 to 30% of people over the age of 60 will, if you look at their blood and you test for mutations, they're going to have mutations that we previously thought were only in in blood cancers, and actually JAK2 is the 5th most common mutation in this clonal hematopoiesis of indeterminate potential. Basically, people just walking around without any blood count abnormalities, but if you look, you can find some mutations in their blood. And we think now that there's a spectrum of going from what's called CHIP to MPN. With CHIP, you have a very small percentage of mutant cells in your blood, and as they grow to a certain level, then you develop blood count abnormalities, for example, leading to a myeloproliferative neoplasm. And so, moving back to what is this familial predisposition to acquire a myeloproliferative neoplasm? We know the JAK2 mutation was not acquired, but are there other genes in our body that we have acquired from our parents that sort of make our bodies a little more likely to acquire a JAK2 mutation? So from this study, it's actually from the 23andMe data. So when you give your saliva to 23andMe, you click a box that says they can basically do research on the saliva and don't need to tell you. So they screened about 500,000 people's saliva. Number one, screened for JAK2 V617F, and again found that normal controls will have JAK2 V617F, and and that's acquired mutation. But then they looked at their other inborn variants, we'll call them variants now, not mutations because they're not associated with the disease. Are there any other sort of tendencies, the people who had the JAK2 mutation, what do they have in common in terms of variants in other genes? And interestingly, the one that came up to be the strongest was a normal variant in JAK2 that interestingly about 50% of Caucasians will harbor. And that is a very common, if you're going to have a JAK2 mutation with MPN, you're very likely to have this common JAK2 variant. So I want to clarify, because I've had patients insist that they have this is a very common variant. If you pull up your 23andMe data and you can see it, it's a very common variant, but it doesn't necessarily mean that you have MPN, but it just, if you have it, you might be a little bit more likely to get an MPN. Other genes that are coming up, for example, TERT, that's in a telomerase, which are the ends of our chromosomes. Each time our cells divide, our chromosomes get a little bit shorter, and so that helps them sort of keep the ends. SH2B3, interestingly, is involved in thrombopoietin receptor signaling that makes us make platelets, so that makes a little sense too. But again, none of these would make you get an MPN, but just increase your likelihood of getting an MPN. So possibly many of these things together might just sort of make someone a little bit more increased tendency to get an MPN than someone else who doesn't. And this is just a picture. I'll try to explain it. It's a little bit complicated, but we know with a myeloproliferative neoplasm, we have the MPN driver mutation, which is your JAK2, your calreticulin, or your MPL, which gives you the high platelets, the high red blood cells, sometimes the high white count. But when somebody progresses to myelofibrosis or an acute myeloid leukemia, they usually develop additional mutations. And many patients just in a chronic phase of ET or PV can also have additional mutations. And some of these mutations are valuable to know because they may help us predict whether somebody is going to progress more rapidly or not. But we think that addition of additional mutations will sort of move your MPN from just your chronic phase to more like a leukemia phase. And so then this brings into how to interpret these NGS studies. So NGS is next generation sequencing. So many times patients, particularly on their bone marrow biopsies, as part of the bone marrow biopsy will have sequencing which will include your JAK2, but will include a lot of other a lot of other genes. And many times these panels can be quite large because they're used not only for myeloproliferative neoplasm, but other cancers. So one panel, and they just want to put all the genes that are associated or have any relevance to any cancer on that. So you get a lot of info. Can also get info not only about your acquired mutations, but sometimes accidentally can get info about some, whether you've acquired from your parents some mutations that we know or sort of increase your risk of cancer. So on these reports, you may notice different tiers. So usually on the top will be pathogenic mutations, or tier 1, mutations that we know are associated with a disease and are known to be acquired. For example, a JAK2 mutation will be up there because we know that this mutation is clearly associated with disease and it's important. Then lower down, many patients, because everybody or many people, everybody is a little bit different. We don't have all exactly the same genes. Many times people will come up with multiple variants of unknown significance, and they're just that. They're variants that are not necessarily the same as what's defined as the reference, but that particular variant is not has never been described to be associated with a disease. So do not know whether it has any clinical relevance or could potentially just be potentially your family's personal variant that really has no impact. And many times on these panels, just want to clarify, in particular for the variants of unknown significance, can't really tell if you just got it from your parents and that's just your family's variant, or at some point in your life you acquired that mutation in your blood. So I think on these panels, the most important things to focus on would be the ones that are listed as pathogenic, and maybe for now, just leave things that are listed as variants of unknown significance just at that unknown significance, and you can pretty much ignore them. We are performing a myeloproliferative neoplasm family study, which is taking a little bit of a different approach. Instead of wanting to know what was inborn in the families, we're wondering whether the unaffected families may also harbor blood cells with mutations, but not necessarily as strong as the JAK2 mutation. For example, may think that to have an analogy, a JAK2 mutation could be like very hot, like a Goldilocks principle. A JAK2 mutation could be really hot. Maybe, and it causes a problem. Maybe there's patients or people in these same families that have acquired sort of a lukewarm mutation, that it's sort of similar to JAK2, but it doesn't cause them any problem, and they're just basically hanging out with these sort of lukewarm mutations that are no problem whatsoever. And so to do this study, we wanted to do, because MPNs are quite rare, so figured that we need to do this study by mail, and so needed to get samples from patients or people around the country, and wanted to do it in a way that was sort of non-invasive, could be done at room temperature, are shipped at room temperature, and can be collected in the people's at home. So basically making it as easy and cheap as possible. So a student in my lab named Eli is really interested in developing non-invasive ways of studying mutations in people's blood. So turned to saliva. So you may not know it, but most of our saliva has lots of white blood cells in it. That's how the 23andMe was able to easily pick up JAK2 mutations, because when you spit and you give them a saliva sample, the cells that they're really getting out of that is actually white blood cells. So in this, he just wanted to, in a normal control as well as an ET patient, just took blood and saliva and just demonstrated by different methods that they looked basically very similar in terms of the white blood cell content. And then in a panel of patients in whom we had their blood and they basically spat in a tube at the same time that the blood was collected, we did mutation role profiling basically on the two samples and asked, are we getting the same results? And we did get very similar results both for JAK2 as well as for patients who had additional mutations. So could really pick up the same mutations in the person's saliva as in their blood, and even mutations that were only a small fraction of their blood, we could also pick up in the saliva. So this is just demonstrating that saliva can really be used as a surrogate for blood that's non-invasive and can be shipped at room temperature and easy to collect. So this is sort of our concept of why we're wanting to look for mutations in MPN families. We now know with this chip where people get mutations in their blood, many of the mutations that are very, very common in just the general aging population, such as in TET2 or DNMT3A, those mutations make the stem cells resistant to chronic inflammatory stressors that we all see as we age. And we also know that when people have specific sort of stressors, such as chemotherapy or radiation, they have outgrowth of cells with different mutations that render them resistant. So, like trying to think of your blood stem cells like as evolution, because that there's a pressure, and then the cells that are resistant to that pressure will grow out. So similarly, in MPN patients, we think that there's a specific type of inflammatory pressure present in the MPN families for which cells like JAK2 will be very resistant and grow out. So we're wondering if there are, what other mutations can be seen in these families. And so we have this is just our recruitment. It's a remote study, and patients or people can do it all through the mail. We've tried to do it as sort of economically as possible by using just the plain U.S. Postal Service, doing everything in room temperature, and it's very easy for an online consent form, very easy for people to participate. And so to begin with, we took a unique opportunity where we have some discordant identical twins, basically one with an MPN, one without. In this case, we have a few sets, but this one actually has a set of younger men who have, one twin has calreticulin mutated myelofibrosis, and the unaffected twin is unaffected. And so in the affected twin, he has calreticulin, but interestingly, in the unaffected twin, we found, who is perfectly fine, found a mutation in JAK2 that not in the usual place, but a little bit different place and not necessarily ever described in a disease, as well as a mutation in MPL, again, not in the usual location. So what we're doing now is asking whether these two mutations can sort of slightly, our hypothesis is they're sort of slightly increasing signaling like a JAK2 mutation. So our concept of they're like, it's not too hot, it's just a little bit, these mutations may be a little bit lukewarm, which are not really causing the unaffected twin much problems. So we are looking for participants for our MPN study. So if you have one other, and we're defining an MPN family very loosely as if you have one other MPN patient in your family, or you have MPN and there's a second patient with any other sort of hematologic malignancy, we'd like to basically very, very loose criteria, because we're really interested in everybody, and we're interested in the affected and unaffected family members as well. So thank you.